We're talking about DNA repair, the SOS response. When DNA is damaged, oftentimes mechanisms such as the base excision repair and nucleotide excision repair are going to come in directly to the DNA using enzymes that have already been produced, such as nuclease or a DNA helicase, and fixing the DNA as simply as they possibly can. The SOS response is a little bit more involved than that. We're going to have translation of genes and a little bit more interaction from the entirety of the cell. So it becomes pretty clear by the end of this why this is called the SOS response. Here is the setup of our cell, very elaborate. We have our DNA ready to be replicated. Next to it we have inactive Rec-A bound to its repressor. And then down here, these lovely green blobs on top of the other strands of DNA. Those are representing Lex-A, which is going to be a repressor binding to a 20 base pair consensus sequence that we're going to call the SOS box. Lex-A has different affinity for the SOS box depending on the genes that are surrounding the SOS box, and that's going to come into play a little bit more as we move forward looking at translation of genes down the line. In order for the SOS response to be called upon, there has to be some kind of damage to the DNA. This can be UV irradiation, alkylating agents, pretty much anything can initially trigger this, but the ones that really tend to need it the most are the ones that have the larger effects on the DNA structure in. We zoom in a little bit closer here, and that interesting pentagon is going to be representing our DNA polymerase. As it's moving down the DNA, it comes across there damage here and can't get past it. So we now have this accumulation behind it of single-stranded DNA that is going to have the same sequence as it tries to get past this, and as this accumulation builds up, we have Rec-A becoming activated and forming filaments around it. As I said, this is our active Rec-A, and we are going to use just a little bit of ATP in this process. Zooming back out to our entire cell, we have active Rec-A available now to interact with Lex-A. This interaction allows for the cleavage of Lex-A, and this de-represses the genes that are downstream of the SOS box. These little pieces of DNA are going to give us a little dance, and proteins are made just like that. UVRD, UVRA, UVRB, there's a couple more that are made as well, but these are considered to be the early proteins within this mechanism. This just means that they had a lower affinity for Lex-A, and together they're going to be trying a little bit of a simpler method. We're going to attempt the nucleotide excision repair in order to fix this. This is nice for the cell if it works. It means they don't have to put in quite as much work as they thought that they were going to. However, if it doesn't work, you're going to see that it takes a little bit more energy from the cell to finish this off. So when it doesn't work, we have a continued accumulation of the single-stranded DNA, and that means we have continued activation of Rec-A, and therefore more cleavage of Lex-A. The genes in SOS boxes that had a higher affinity for Lex-A are now being involved as well. So we start to see our late genes here have some more beautiful proteins. UMUC, SOL-A, and UMUD are a couple of these later proteins. SOL-A stops cell division by binding to FTSZ, and this causes elongation of the bacteria without division, also known as filamentation. After this, UMUDC, dependent mutagenic repair, can get started, but as you can tell, this is a much more drastic process for the cell, and obviously it's the kind of damage that they need to take care of, but at the same time is kind of costly for the cell. So pretty clear what's called the SOS mechanism. Thank you for listening.